Thank you, Mr. Chairman, and we appreciate you witnesses being here and sharing your expertise. So my first question, Dr. Jernigan, deals with, in your testimony, you mentioned the development of a mobile mini lab, a cloud-based platform that can be set in a remote resource-limited settings to process test virus specimens and to send that genomic data up to the cloud for further analysis and action. So could you elaborate on how this cloud-based platform would allow public health officials to address outbreaks quicker and more effectively in a largely rural area like my 4th Congressional District in Indiana?
So yes, I think we were referring to the use of these micro technologies like this one here, which actually, actually is a sequencer. And so you actually take the specimen, prepare it in some little boxes that we take that fit into a carry-on on a plane. You prepare them, and then you just simply inject it in here. There's a way that you can actually do what's called barcoding of the specimens and do multiple specimens at one time. And with that, you get a sequence. And the sequence just tells you the genes of the influenza viruses. So this is something that we have demonstrated in various different settings. We actually did take it to Iowa, to a swine fair, where we actually swabbed a number of the show pigs. And at that, we're able to quickly determine if they had influenza, the swine influenza that was circulating among that group. That data plugs into a laptop through this little USB port. And then on the laptop, it runs a lot of the information and prepares the signal that gets sent up to the cloud, where we have a process called IRMA. IRMA is a tool, a pipeline tool that actually takes the data and uses machine learning and artificial intelligence to try and determine which of the flu viruses are actually in the sequences. That information then gets pulled down by our bioinformatics staff at CDC, where they can then, if needed, generate a vaccine virus. And so this allows us to take the tool to the place where the problem is occurring, rather than having to try and figure out how to get viruses to the CDC.
So to take that one step farther, you could regionalize, or wherever you collected your data, then you could develop a vaccine specific for that areas, which
it's possible.
Quickly, so.
The manufacturing process would let you probably not be able to do that, but yes, you can tailor what you know about in certain regions. I think Dr. Watkins will probably get into some of the data issues in the subsequent testimony.
So you mentioned pigs, and I have a background in agriculture, so when you were swabbing those pigs, any thoughts on the African swine fever, I think?
Yeah, so African swine fever is something that's different than the swine influenza, and so I'm not an expert in the swine fever, but certainly these same kinds of technologies could be used anywhere in the world to do that kind of detection.
Thank you. Dr. Fauci, you have any thoughts on that area?
Yeah, the point that Dr. Jernigan made is interesting. I'm in some respects glad you brought that up, because we constantly get people confused between African swine fever and influenza that's in pigs that could recombine with an influenza to give us a pandemic. It has absolutely nothing to do with that, but sometimes people get confused when they hear the word African swine fever, which is really completely unrelated to influenza.
And I appreciate that. That's part of the reason I mentioned that. So I thank you. I yield back.